hot.
Too bright.
And this other one next to it too, that 22 dB one.
Contrast.
Try turning that down.
I love this question. Someone wants to know what we do for our day jobs, or is this our job every day? I want to talk a little bit about your day jobs. I don't know. Who wants to start? Nicole made eye contact. She has to start.
This is kind of my day job. I come out here usually 2 months a year and get to lead expeditions. It's great fun and get to work with amazing people. And then the rest of the time, my job with Ocean Exploration Trust is chief scientist, which is bringing together teams of scientists to put together these expeditions and doing the reporting on them, making sure the data is getting to databases and archives,
Yeah,
and
it's okay.
you know, making sure we're doing good science with our exploration and ship time. So that's what I do. How about you, Leela?
Oh, my day job is being a graduate student. So I'm a grad student at OSU in Dr. Andrew Thurber's lab. And we do part of what we study in our lab is methane seep ecology. So I really first got exposed to the deep sea hands-on in this way during my Nautilus internship 4 years ago, as an ocean science intern. And since then, I've kept coming out on Nautilus, usually for maybe like, I don't know, a month or a little less a year. And yeah, the rest of the year, I'm a grad student.
Doing research on some of the stuff we collect.
Yeah, doing research on some of the stuff we collect, and some of the stuff we'll be collecting here. So some of the sediment cores we're collecting and the fauna as well to see if they're using seep-derived carbon. So I'm just interested in kind of the role of methane seeps in the big picture ocean.
I have to popcorn someone.
Oh, what's your day job, Megan? Did you already say that?
I
missed that
didn't.
part?
I didn't. No. Similar to Nicole, this is kind of my day job. I come out here between 6 and 8 weeks a year. Love it. Absolutely love it. This is my 9th year of sailing on Nautilus, which has just flown by because it is always so exciting and different and new, and get to meet new people and learn new things. The rest of the time, I'm the Director of Education and Outreach for Ocean Exploration Trust, which means I help organize all the ways that we reach out and share our story with the world, which is our social media, our live feed, our videos, our educator programs, our internship programs. Yeah. How about you, Jeff? Popcorn Jeff, what is your day job?
I remember now.
Well, my day job is not being a navigator for a ship in an ROV, but my day job is a research scientist at Oregon State University and a researcher also affiliated with NOAA's Pacific Marine Environmental Lab. I'm a marine geologist that uses seafloor mapping data to understand the interaction of the deep sea and this geology with the ocean. So in the Earth-Ocean Interactions group, we tie processes that occur in the subsurface to the ocean. And that's mainly why I'm out here is I map and characterize methane seeps. And they're a conduit and a connection between the Earth and the ocean. And I also work on earthquake geology and just in general seafloor mapping of the U.S. waters. So I was just recently out on another expedition, on another vessel, studying the Cascadia subduction zone, which I'm back out on.
Oh, man.
I had about a week break between cruises.
What ship were you on before?
I was on the Marcus Langseth.
Okay.
which is another UNOLS vessel that's capable of deep seismic imaging. So we were out there collecting seismic profiles, looking at the downgoing plate, essentially what is making the subduction zone here to better understand earthquake hazards along the Pacific Northwest.
Do you do those surveys on the inland edge of the accretionary wedge, like through kind of the slope, or do you do them on the ocean side?
Both. We traverse the deformation front from the abyssal plain all the way to, as close as we can get to the coast.
Okay, then you are the perfect person to ask this question. Then everyone else still has to talk about their day jobs. But could a really big earthquake knock a lot of methane out of the seabed? What do you think?
That is a great question. Cascadia hasn't had a great earthquake in a long time. So us going down here and exploring and getting a baseline understanding of how the methane is escaping right now could give us a great opportunity to come out here if a terrible thing like a big earthquake could occur to investigate that. But my suspicion is that there's a lot of shaking that occurs, a lot of movement on the seafloor, and in some areas you could have a significant amount of uplift, which could create new pathways for methane to escape. But we don't know. And that's why we're out here doing it, to try to understand what's the conditions right now.
Awesome. I have about 1,000 follow-up questions. I was asking Tamara a little earlier, what do we know about kind of how big the pockets of methane are here? If there are, someone asked earlier if this is like a tank of methane, and it's kind of like one big, as I with my limited art skills would probably draw this, like a big bubble of methane, and then it's kind of making its way out through cracks, or is it more being generated slowly and being released slowly? Do we know?
Well, there's somewhere a reservoir of methane that's being tapped, and we have these leaky spots. And there could be a deep connection where there's a sedimentary unit that's porous, that has the ability to have methane in it and hold it. And then with the migration pathways, it's kind of bubbling out on the leaks of like a leaky faucet or a leaky pipe system. Or you could have that would be a deep reservoir. There could be shallow reservoir too, under the carbonate. Could be accumulating these bubbles, and they fill the void spaces in the carbonate, and then they find their little cracks through. That's why possibly we see it kind of in many different spots along these carbonate ridges have seeps, and they're transient, where they happen for a while and then they move to another spot. But evaluating any one kind of reservoir of methane or a pool is, you can do that with some imaging techniques or with the geochemistry. It's like, how deep did it come from? What are the sources of it?
Do you want to popcorn someone else about their day job?
I don't know, Mike, what's your day job? This
has been my day job for 9 or 10 years now. But I recently started doing another day job. I started doing marine spatial planning work in Labrador with a Innu Nation organization, which is a representative body of the Innu people in Labrador who are indigenous there. So there's 2 communities. And we're starting our efforts partway up the north coast in the community of Natwashish. And I just started it like it was just about 8 weeks before I left. So I'm kind of doing it in a little bit of a part-time role right now. And we're all gonna take it up in earnest there in the fall. So this is the turning point for me where this will be a part-time job for me in the future, I think. But I still hope to get out here a couple of times a year.
What's that little orangey yellow thing?
Yeah.
Able to zoom on it quick?
Zoom in there, Tammy.
Little anemone.
Anemone.
And that's like just a piece of carbonate, I suppose, behind it. Very nice.
Yeah, nice pirouette.
All right, full wide. Catch up a little bit.
All right, Randy, what do you do for your day job when you're not here on Nautilus?
Yeah, I'm similar like Leela in a sense. I'm a graduate student. Um, Now it's going to my fourth year of my graduate degree. What I mostly do is I'm in a writing phase right now, so I had some data. One interesting thing is I actually had data from Herc and how loud Herc actually is. So we did some analysis on how loud Herc is at 10 meters, and apparently it's really loud. So more exciting stuff to come is one thing that Brandon Phillips is actually developing in his lab is a very silent camera system and very small, so it's not very loud, pretty low light and all that, and that's what we're interested to deploy and actually see like how effective it is or how does it make like fish not run around so much. So yeah, that's much more of what I do, and this is my first expedition on Nautilus, which is such a great experience.
Do you recall how many decibels Herc is at 10 meters?
Yeah, so the loudest thing, which was pretty interesting, is the HBUs, and that was around almost, well, hold on. At 10 meters, we heard it was around 90
Oh,
90
yeah.
dB. No, much more than that, around 110, 120.
Wow.
That was around 60 hertz, so it's definitely the HBU motor. And also the USBL pings actually were but they were at such a high frequency, right? You can hear them pretty loud. If you can hear at that frequency range, they're pretty loud. They're around similar 90, 100 dBs around.
On this cruise working with PMEL, in the last 2 years, we brought a hydrophone that we deployed, a small one, or they supplied one, and they actually left it at a seep, and we transited away from it by, I think, a few hundred meters at least. You were able to pick up the sound of the seep bubbles, but you also had still the sound of the vehicle in the background.
And what's funny is that is the exact same data set that I have.
Bob
Gotcha.
Ziak.
Bob Ziak, yes. He's writing with me on one of my first papers.
Oh, that's great.
Yeah, Bob Ziak.
Yeah, so the bubbles make a little bit of noise,
Yes,
but
yes.
definitely not as much as Hercules.
Yes, that's true.
Yeah.
It was a great paper that Bob wrote about that cruise, about the bubbles.
Well, it's interesting to think of all the things you can do with passive acoustics and being able to relate it to seeps and how frequently they seep. That's one of the questions that, since we only watch them for a short period of time, that scientists still have in a lot of cases. We know from mapping over the same areas at times that they're not always active. But if you put a hydrophone down, you can listen for when they are active and
That is true.
have some more information on that.
Pulling up 2 meters.
Lots of nice welcome to Nautiluses for you, Randy and Mike. Lots of nice, can't imagine Nautilus without yous, and congratulating you on your new job.
Well, I hope they don't have to carry on without me.
Yeah, we need you back.
I should have. I ideally want to keep my hand in, and with other ROV groups I work with too, but especially here because it's just such an awesome group, a lot of fun. So I hope I can get out a couple of times a year.
All right, Tammy, you're up next. Shannon will anchor us.
What am I doing? I stepped out for a second, so.
The question was, what is your day job? So we're discussing if this is your day job or if you have a different day job.
Oh, that's kind of a loaded question for me. Here, what don't I do, really? This is my summer job, I guess you could call it. I do work with other video systems on other vessels as well. I run a small portable system for Ocean Networks Canada, as well as in the off-season, I do a lot of the documentation and inventory for Nautilus's or Ocean Exploration Trust video system. So I'm pretty much in charge of most of the video system side inventory, as well as I'm a personal trainer. So that's kind of my other half of the year full-time job, I guess. And I'll obviously still do some kind of programming or emailing with clients while out here. And I recently just started to kind of, guess we could call it just be a professional organizer. I do a lot of organizing or little creative work for different people, different organizations, just as needed. It can be anywhere from, hey, I want to start this Etsy page. I don't know where to go, and I have all these projects. Which one do I put first, or how should I track it? To, I just have so much going on, and I need someone to help plan out my day and make sure I stay on track to things, as well as just doing social media. I think that's pretty much it. I used to work in restaurants here and there, have a few connections with people I know that when they need help, they'll just kind of call me up and I'll go in and serve or bartend, catering, different things like that as well. So it's nice to have a flexible schedule, but also I have an issue with saying no.
I was gonna say, you have the overachiever of day jobs. You have all of the day jobs. Awesome. You really are the reason so many things here are organized and labeled. It's fantastic.
Yeah, it's always funny when I'm somewhere else or on a different ship and someone emails me asking where something is. Even if I haven't been on Nautilus for a couple months, they'll be like, oh, it's...
Have you checked here?
Yeah, it's in this bin, probably in the back left corner of the bin. Just have one of those memories, I guess.
Check out the Argus view right now.
Yeah, it's so good. I was just looking at that.
Yeah, I was just going to say, can I zoom in a little bit, Randy?
Sure.
Yep.
Yeah.
Get a little bit of a...
Mesmerizing.
Like, oh, I want that Argus capture to turn out.
I know.
See if I can get
Past the siphonophore.
Yeah, get the siphonophore out of
Get
there.
out of there.
They're always crashing the photos.
I know, always. Siphonophores themselves. Wow, so cool.
Yeah, so many. All right, Shannon. Tell
I
us about you.
I just started recently my day job, I guess you would say. I am a lab manager and kind of research scientist at a lab at the University of Washington, but we're associated with NOAA's Pacific Marine, PMEL laboratory. So I'm a NOAA contractor and I'm in a genetics lab. So I'm on the boat to filter samples for eDNA. So I just started the job 2 months ago. I only graduated with my master's in marine science last year, so I'm excited to continue learning. And I've never done something like this before, so it's really fun.
Was that in the job interview? We need you to be on a boat in 2 months after starting.
By the way.
No, it was like the day after I was hired, I got an email, hey, would you like to go on a cruise? I'm like, well, absolutely. Yes, please.
Now, did they say cruise or expedition?
Oh, yeah.
There's a hot topic.
Yeah, it was probably an expedition. I'm so used to smaller vessels. So when it's, a tiny little boat that you dive off of, it doesn't really feel like it counts as an expedition, but the size of this vessel and the work that gets done definitely classifies it as expedition.
And you were saying you did more, so you mentioned diving, you did more shallow water work before, right? Or
yeah, most of my work has been intertidal or diving related, so, only 30 meters down. And most of it has been in the tropical water. I did a lot of work in the waters around the Middle East and in the Caribbean. So this is the deepest and coldest I've been in a very long time.
What were you studying when you were doing the dives in those regions?
I am a worm nerd. So I really like this class of worms called sabellids. Oh. They are feathered ester worms. I think they're beautiful and great. Not everyone does. But both my undergraduate and graduate research was focused on them. So I really couldn't escape.
No, they're awesome.
That's okay. I'm a sea slug nerd, so.
I support that. I support all invertebrate nerds.
yes, invertebrates are so cool.
They are.
My undergrad project was on nudibranchs in Hawaii, so.
You picked the prettiest things in the prettiest place.
Seriously.
But I concentrated on the sap-sucking sea slug, which isn't quite nudibranch, but they're very interesting.
Pulling in 2 meters.
Roger.
Well, we have just about an hour left of this dive. How far have we made it, Jeff, to waypoint 7?
We're doing pretty well.
Yeah.
Is it 6:30? 6:35?
It is 6:35 right now.
I'm not sure if we have quite an hour left, Nicole.
Yeah, that's fine. It was a
I think we were
rough
saying
estimate, right? What do you think?
we
Mike,
could recalculate, but I think we were talking about off bottom quarter after.
Okay.
But we could.
Yeah, Bob was saying something 15 meters a minute on the way up.
Yeah, that's all we usually hope to get.
Yeah. So what do we have left, Jeff?
Well, if we were trying to get to the finish line around waypoint 7, that's 875 meters.
We've made it about halfway.
We have.
I think we can do it. Jeff, when you were in the lounge earlier this morning, you seemed like the type that wants to meet those goals. So I have a feeling we're going to make it.
That's right. Well,
like take 800 meter step bearing.
Yeah, we'll take the bearing and bump up the knots. But no, there's something about completing a dive plan that's very fulfilling. But the waypoint 7 was a stretch goal.
Okay, I feel like this watch team could get us there.
Well, if you're really hell-bent on it, we can go faster. Well, you don't get to look around as much.
Well, the fish are leading the way, and they're also
Yes.
occluding everything.
Yeah.
No,
Are we hurting the fish or are they everywhere?
Yeah.
They might be
That's a real
hurting
question.
us.
Good
Yeah.
question.
Oh, that, are we in need of samples?
Discussion happening. Don't worry, viewers. Our dive plan for this entire expedition. Ooh, good Argus capture
Wow.
again. Yeah.
There we go. Nice.
Captures. Awesome. Our dive plan and dive tempo is going to be to launch at 8 a.m. Pacific and be back on deck by 8 p.m. Pacific. And we will map during the night, dive during the day. The team will also be processing samples in the lab during that time. So that's the tempo we're picking up starting today.
Very civilized.
Yes, it's very, very nice.
So the discussion back here is about whether we try to take push cores at some point, and this does not seem like a good spot to stop and try, but I suppose before the end of the dive we might, if we come across another area that looks like it has a little bit more sediment.
Sure.
Have to push the fish out of the way.
Yeah, you'll get a piece of fish if you don't watch out.
Fish sticks. Okay, Nicole, question for you. So since you've been doing this for such a long time, what is the coolest thing or strangest thing you've seen over the years?
That's probably my least favorite question, and not to say that it's not a fair one. It's just there's so many cool things that we've seen. It's hard for me to pick. Certainly the unexpected things. I mean, coming across a whale fall while we were already in awe of all the octopus that we were seeing was really, really neat. I had a similar experience in the Med back on, I think, my second expedition with Nautilus. We were on Arethasthenes Seamount surveying for sinkholes that were on the top of the seamount, and we came across a giant shipwreck, ancient shipwreck that had, most of the shipwrecks we found in the Med had small amphora piles, and that's how you identified where they were because the wood had all deteriorated. And this had these massive pithos, which were huge cargo containers, clay cargo containers from a ship. And so that was quite unusual and unexpected. On that same dive, we came across a fossilized whale skeleton. So it was just a very memorable dive.
No kidding.
How about you? You've been out just a couple years shy of my record, I think.
You know, this question is also so hard. It's like superlative question. What is the most blank, most crazy, most... And I think my answer changes like all the time.
It's like picking a favorite child or something. I don't know.
I know. Strangest or coolest? Gosh, I just, I I'm going to like go back to biology probably. It just some of the animals and some of the adaptations that are here on the seafloor are incredible. You know, the fact that a googly eye stubby squid is a real animal is a pretty strange and wonderful thing. Cleverly disguised as like a kid's toy, you know, but an animal totally adapted for the environment.
The gulper eel was like
Gulper
that to
eel,
me too. That
right.
was like something out of the Muppets or something.
Exactly, exactly. I don't know, and then I think, you know, some of the coolest ones are probably just stuff that I learned about on the fly, you know, like the first getting to visit hydrothermal vents, like in having never, you know, I have a biology and chemistry degree, but not like a marine geology degree. So, you know, that was all new. And
Even so, seeing those for the first time, like you said, there's spectacular.
Like, yeah, you're like, this is 10 stories tall and 400 degrees, you know, so churning out of the top. And then also some of the archaeology is really memorable. I think USS Independence is one of my favorite all-time archaeology dives that we've done. That was an intentionally scuttled World War II aircraft carrier off the coast of California. And that one was cool for lots of reasons, including being surprised by having a plane on board that we didn't expect to be there after all the research. As we did the website overhaul, we talked about doing a like staff favorites pick, you know, so that we could have kind of different themed things in the gallery. And I think we decided to implement it, but it was like one of the hardest meetings because we started talking about like who would get to pick the staff favorites and like how often they should change because, you know, it's such a long list of favorites. And we were like, this needs to be revisited, you know.
Well, it's like browsing in your favorite bookstore then, where they have this staff picks their favorite books. Seems like a neat idea.
It'd be fun. We could like shop it around. Everyone gets like a month of like, now it is Nicole's favorite picks. It's March, you know.
Anyone else who's sailed have other favorite
cool
superlative moments?
Yeah, the whale fall is memorable, and the octopus garden, which is kind of it's in the same place. But that really neat. I guess it was a giant, kind of a squid thing that Gabby was flying. It's all over the highlights now. That was pretty neat. When we did the seamounts with DFO, I think in 2017.
Yeah, I was
the
just
Bowie one.
Delmont seamount. I was just gonna say that.
That was amazing stuff. Yeah.
Oh, that like Spongetopia or whatever you call it.
Super clear water, ridiculously large sponges. The yoga pants sponge.
Yeah.
All excellent choices. I really liked the Gulf of Mexico coral dives too, you know, and I think that was just like the science objectives were so cool because it was like sit down super close in front of this coral and then just like zoom the cameras in and look at them in, you know, intricate detail, and they're kind of jewel toned oranges and pinks and yellows. And so we got a lot of really great looks at corals.
Well, and looking at the shipwreck in the Gulf of Mexico, that was one of the only excavations we've ever done. So it was really rare for us to get permits to collect archaeological artifacts.
Oh, yeah.
We were working with some teams from Texas A&M and NOAA to excavate the shipwreck that had been discovered earlier but not fully surveyed. And so we did a complete photomosaic and map of it before we started excavating, and then we were able to take some artifacts so that they could try to date it and tell where it had come from. Then we found 2 other shipwrecks right nearby, so that they think that all 3 went down in a storm. kind of powerful to be out there in the middle of the ocean and realize just the power of the ocean, I suppose, and what what can happen in big storms like that.
Zoom in there, Tammy.
There have been a lot of neat things in the water column on this dive.
Yeah.
Yeah, what a great shot. Are the threads off of it, are they mucus or are they stinging cells off of this?
On some there's stinging cells, I don't know.
What great flying, Mike. Great shot.
All right, that might go up forever. It might go to the surface. Pretty cool.
Really cool. All right, Nicole, what do we know about siphonophores? Give us some siphonophore facts.
They're colonial organisms. So but they each end up with like independent functions, right, that help. So they're kind of a great example of teamwork because everyone has their own job, but they all come together to make this cool, massive organism. What do other people have for siphonophore knowledge?
Teamwork makes the dream work was...
That's as good as we're getting, huh?
Yeah.
Yeah, you nailed it. I mean, the individual ones are called zooids. That's what I've got. I don't have a lot more siphonophore knowledge. You know, some become digestion, some become defense, some become locomotion specialists, flotation specialists, buoyancy. But yeah, you nailed it. Maybe if we have some siphonophore experts in the chat.
Well, some of these things you think how difficult they must be to study. I mean, the midwater is, well, we know the least explored ecosystem on the planet. So we focus on the benthos with the ROVs, but we're definitely interested in being able to study the water column. It's this vast ecosystem that, you know, is so important to our planet, but we really don't have very many tools to do it effectively.
Yeah, the Schmidt team's giant siphonophore sighting that they found in the midwater down off Australia, you know, and same type of animal, different species of it, was in a big, you know, curly Q line, flattened out,
What?
a big spiral ring, and they,
you
know, approached it just right with the ROV that they didn't fly through the midst of it. You know, they got this incredible view.
Yeah, and you know, because these things are so infrequently observed, scientists can do a lot with footage like that. There have been scientists who've published papers based on like 30 seconds of observation of a deep sea fish. So that's why what we do is really important. You never know what an expert out there is going to be able to learn from the footage that we get.
I don't know that it was a new revelation, but it was a new revelation to me when we saw those blobfish defending their eggs in pairs. And I was like, blobfish are great parents. I didn't know that. Way to go, blob sculpin, you know. That Argus shot is so cool again.
Yeah, that would
And I
make
just
a
know
great
that capture.
screensaver.
I know that capture like won't look quite right, but yeah. Squad of sablefish. Jeff, you want to describe where we are on the feature now? It's, you know, kind of silty sandy again, sedimented.
We're the overall ridge that we've been on. We're about at the midpoint, which is a low between two highs. There's a southeast high spot that we were going to get to if we had we reach waypoint 7. But this is we've been traversing just a pretty low gradient, flat top of this ridge. It appears to be pretty, has a nice sediment cover, at least a thin one.
I know, do you think it's worth trying to see if it's coreable? Seems like a lot of rubble still.
I'm not overly confident.
Yeah, I that's why I haven't stopped
either.
this yet, because I don't think it
I haven't seen any spots that look a bit better, but
no, I think we should just keep going for now. So the fish have been with us since that high spot, since the knoll.
yeah.
Yeah.
We
How far away from there are we now?
We are from roughly 700 meters from that high spot when we came up on it at waypoint 6. It seems as like the fish schools are on top of the ridge. And where we saw the seeps on the northwest edge of the ridge, not as many fish, but much more active seepage going on up there.
I really want to know how many of these sablefish are escorting us and how many new sablefish we're encountering. My sablefish fun facts here that I found, the juveniles can migrate 2,000 miles in their first 6 or 7 years after settling out. So,
wow.
You know.
Long distance swimmers, endurance.
Exactly. Not a huge leap to think they followed us the last 700 meters off the high.
Well, and particularly the juveniles are vertical migrators as well. So they're more likely to get eaten by like birds or larger fish. I'm not sure how much vertical migration an adult does.
Yeah, it's a good question.
Because they probably, as an adult, they probably get enough fish and are able to eat more of what they find in the deep.
There's that screensaver shot if you're watching on channel 2.
Yeah.
It's the best one yet.
When reviewing the multi-beam data that we collected last night, the ridge to the south, which we thought was going to be our dive site, we were picking up a lot of biologics in the water column, a lot of what is usually considered fish balls.
That's right, you mentioned that.
Multi-beam can act like a very expensive fish finder. Think of the sound waves moving through the water column with many, many fish through it. It quite easily picks it up.
What does it look like on the returns?
They're high amplitude, and usually it's attributed to the swim bladders in the fish. So each one of fish that has a swim bladder is like a bubble swimming around. And as we've talked about earlier today, the acoustics are very good at picking up bubbles. And these fish can be large schools of bubbles moving around the water column.
Mobile bubbles. Yeah.
They are in a way, and since they usually work very close together, they kind of are an amorphous ball in the water column. They're quite different than seeps, which are vertical and narrow, whereas the fish balls are kind of broad and amorphous, and they don't kind of have a vertical structure to them.
I'd love to see. Is that saved somewhere in our data?
Yeah.
Could I see an amorphous fish ball data return?
That will be when we review the water column data. It'll always be.
Oh, you're super quiet.
Oh, sorry. When we review the water column data, it'll be there, so we can get some screen captures of it.
Awesome. I want to put it side by side with one of these photos and be like, when you get this, it means.
Yeah, this is what you might be looking at.
Yeah.
Well, since we're here to study seeps, one of the interesting things to me is that the fishermen actually reported seep locations early on, like commercial fishermen and their sonars that are meant to find fish. So a lot of the early papers that reported on locations of seeps on the Cascadia margin were initially reported by commercial fishermen.
That's so cool. So would a fish mass like this totally mask a seep, when we're scanning over that? Do you get both? Do you get your kind of like campfire smoke, seep bubbles, and a fish mass, or are you just, do they kind of shadow the seafloor?
They can obscure
the
seafloor.
Yeah, it would kind of depend on the dimensions of the fish ball because because the seeps can already be tricky to detect, particularly if they're in the outer edges of the swath of the sonar, you'd need the seep to rise above the fish ball a significant amount probably to know that that's what it was and not just part of the fish ball. Or see that connection to the seafloor.
Pay out a couple of meters, Rendy.
Pay out a couple of meters.
This is a time check. It's 7:01 local time. That.
Do you know where your fish are?
About 15 more minutes before ascent.
Yep.
See any coring potential?
Still looks very rubbly to me.
Yeah.
I'm not opposed to trying at least poking at the sediment because we're not going to make that much more distance. So Mike, do you want to just give it a poke?
I can. Do you want to just get past this stuff
Yeah.
here? This
Yeah.
definitely
Every
doesn't work.
time we think we're gonna maybe stop, a big clump of carbonate rubble comes up. So yeah. No sense in poking the rocks.
No, that wouldn't be very effective.
These guys are making a mess too.
Well, the fish are clearing away all the sediment. That's what they're doing.
You want to try right here?
Sure.
If there ever was a spot, this looks not too bad.
Yeah.
So do we need to stop the ship? Or?
Yeah, stop the ship. Yeah.
Bridge nav, can we have a vessel all stop, please? Vessel stopping.
Okay, Randy, probably bump your camera down all the way. Maybe you'll be able to keep Herc in the frame.
Roger.
Okay, can I see the arm with the bubble camera?
Oh, there's a fish poking around.
That's all right. So Randy, just in general, whenever we stop like this, we've been on the move and everything like that.
Yeah.
So then I'm doing this now, right? And it might be prone to going in a bubble. So
Yeah.
you want to watch the ship's position and Argus's position and everybody's position, because you're still swinging in a tiny bit, but we're only at 400 meters, so it's not a whole lot of that.
Okay. Roger.
So, you know, when we make a change like this. Can you go to the sample salvo?
Roger. Sample tray out.
No, the sample salvo for
sample
the
salvo.
monitors.
Oh, roger.
Yeah, and then just get this port one changed to the other starboard camera.
Roger. Starboard biobox camera.
Okay. Hey, up out of there. Bad grip.
No, I think.
Anywhere here?
Yeah, anywhere that looks good to you.
Okay, zoom in a little there,
Try.
Tammy, please. It's good.
No, it's. Oh, yeah.
Not at
That's
all.
about what I was expecting.
Oh.
All right.
We could help. We could make like a clank. No.
Yeah, I guess we all knew that. Let's have to see.
Was worth the try.
Yeah.
Mm-mm. Yeah, it's not going to be anything you guys can work with.
Okay, Nicole, we've got some viewers who are following along with our data feed, and they are curious why they might be seeing the oxygen drop over the last hour. Now around 12. What might be causing that? Is that normal? You know, what is a normal oxygen concentration?
Okay, box in, Randy, if it's
Yeah,
out
that's
at
a
all.
really good question. Oxygen will vary throughout the ocean, and I mean, we were studying an oxygen minimum zone or a hypoxic anoxic area just off Santa Barbara. This area has some concerns, I think, for low oxygen as well. There's a lot of ocean acidification studies and things to look at ocean chemistry out here. I couldn't say whether this is normal for the area, so I don't know. But I'm glad people are making use of our new graphs that we're putting out there so that they can follow along and make these observations with us.
Do you remember what kind of concentrations were we seeing in Santa Barbara Basin?
Yeah, we were seeing basically zero,
Go on the move again?
no oxygen. It was below our detectable limits.
So we got a ways to go.
Bridge nav,
there.
We can resume 0.2 knots on the same bearing.
Yeah, so for those just joining us, we've had the sablefish with us for a couple hours now. These are sablefish, an important you know, an opportunistic predator fish here, a major target of the commercial fisheries of the west coast of the United States and Canada. Part of, you know, the largest groundfish catch in the west coast is in Oregon, and sablefish make up a significant portion of that groundfish catch. They're caught by trawl, pot, and line. And, you know, to the question specifically, like, why are they here? I don't think we have a great answer for that, but we're just observing that they're here, and it's pretty cool to find them along the same feature where we're seeing these methane seeps in this complex ecosystem.
Randy, bump your camera up a little bit.
Oh, yeah.
Yeah, we know that fish tend to like hard grounds. It's good habitat and good nursery grounds, things like that. So could be related to those reasons. That was actually, you know, kind of an ancillary goal of this dive in particular. Most of the seeps we're going to look at are just in more sedimented areas. They won't have as much carbonate, if any. So being able to assess what type of fisheries are out here, or fish, if there were any. So this was, you know, unexpected, but at the same time, of all the sites, this would be the one where we would expect to see fish.
And boy, did we.
Yeah, and boy, did we. Now, if anyone has a count, anyone at home who's been counting.
Don't ask, someone will.
Send us your numbers.
Send us your numbers. Also, don't blink.
Yeah.
Make sure you don't count the same one twice.
I was like, that is a skill. That is a thing I did in my career. This would be extremely hard to count them this way.
Well, those fish are determined to
ships move in if you know this.
Ruin our last views of the seafloor here.
They really are. What is the largest biological sample that we collect? What's the biggest thing that would fit in the bio box?
I think it was actually would be, oh, well, there's the giant tube worm from Galapagos.
Yes.
That we took up in the manipulator arm. But some of the, very rarely we'll take a full coral, deep sea coral for special studies where scientists